Good morning to all of you. Today appears to be a sunny morning, and all of you are enjoying this online class, remaining at home at your leisure time. You are attending. So, yesterday we are discussing on, or in fact, we have completed our discussion on the cytoplasm as well as the cytoplasmic organelles. So, today we will move on to the last component of the cell, that is the nucleus. All of you know the nucleus is a very important component of the cell, and in fact it is also known as the control center of the cell. Why it's called as control center of the cell, we will discuss that later. But now first we will try to understand about the nucleus, its composition and the function. Probably in the end we will be in a better position to justify the nucleus as the control center of the cell. So basically the nucleus is the repository of genetic materials. It is the repository of genetic material for the simple fact that the genetic material, what we call the DNA, is found in the nucleus. So that's why it is called as the repository of this genetic material. It directs all the activities of the cell. In fact, whatever the activities that are being carried out by the cell, they are under the influence or under the direct control of this nucleus. If you take the cells, majority of the cells have single nucleus, what we call uninucleate, single nucleus. But there are some where you can see more than one nucleus. Probably a good example for this organism having more, or a cell having more than one nucleus, is the paramecium. If you recollect your study of paramecium in your first semester, you know there in the paramecium there are two nucleus. One is the macronucleus and another one being the micronucleus. So that's why it's a binucleate. And there are certain cells where there are no nuclei at all. The RBC of the mammals, where there is no nucleus at all. And this nucleus, inside it is having another structure, what is known as the nucleolus, which is the region of intensive ribosomal RNA synthesis, where the synthesis of ribosomal RNA takes place. About that we will discuss later. Then the question is how the nucleus gets separated from the cytoplasm. The nucleus becomes a very distinct entity within the cytoplasm because the nucleus is surrounded by a phospholipid bilayer membrane, what is known as nuclear membrane. There is a separate membrane for the nucleus which makes it distinct within the cytoplasm. So that means the nuclear envelope is the outer boundary for the nucleus. Then on the nuclear membrane you can see number of pores. They are known as protein gatekeepers. About that also we will discuss later. The nucleus is a membrane bound structure which contains the cell's hereditary information because it contains the genes or DNA. And having this hereditary information and DNA, it controls the cell's growth and reproduction also. Probably it's not wrong if you call that the most prominent organelle of the cell. The nucleus is the most prominent organelle of the cell. If you look into the nucleus, externally I have told you the nucleus is externally the nucleus is covered by what is known as this nuclear envelope. Inside the nucleus there will be again a liquid fluid material here. Sorry, where is here, that is what is known as the nucleoplasm. Within the nucleoplasm you can see the nucleolus. Externally it is bounded by a membrane, a double phospholipid bilayer which you can see here, that is the nuclear envelope or nuclear envelope membrane. Inside you have the fluid material which is popularly called as the nucleoplasm. Within the nucleoplasm one can expect the nucleolus. This nucleoplasm is nothing but the cytoplasm of the nucleus. Nucleoplasm is nothing but the cytoplasm of the nucleus. You call it as nucleoplasm. This nucleoplasm and the cytoplasm, they get separated because of this nuclear envelope. Now, coming to the study of the nucleus, especially the structure of the nucleus. If you want to study the structure of the nucleus, interphase is the ideal stage to study the nucleus, because in a dividing cell or when the cell is undergoing mitosis or meiosis, nucleus becomes disintegrated because there will be disappearance of the nuclear envelope. As a result, there will be mixing of cytoplasm with the nucleoplasm. So the nucleus loses its separate identity during the cell division. So that's why in a non-dividing cell, when the cells are not dividing, probably that is the most ideal stage, and that stage is known as interphase to study the nucleus. Now within the nucleus you can see the following components. I have told you what are those components. One is the nuclear envelope and membrane, second one being the nucleoplasm, third one being the nucleolus. These are the three important structures. The nuclear envelope and membrane is very important because it surrounds the nucleus and makes it a distinct, separate, unique within the cytoplasm. So this nuclear envelope or membranes are double membrane structure. There are two membranes and both the membranes are made out of phospholipid bilayer, as you have studied in the plasma membrane. Outer membrane, out of the two membrane, outer membrane is continuous with endoplasmic reticulum, because endoplasmic reticulum is considered as the highway of the cell. Whatever the things that have to be, that comes out of the nucleus, it is carried by this endoplasmic reticulum. Outer membrane is continuous with the endoplasmic reticulum. Then on the surface of the nuclear envelope there are pores and openings, very minute openings. They are known as nuclear pores. These are the minute aperture present on the envelope. These apertures or the pores are very important because they enable the movement of the material, they enable the movement of the molecules. But these pores are highly regulated openings. These are highly regulated because each nuclear pore is guarded by or regulated by several protein complexes, and these several protein complexes acts as walls. So they regulate the inflow as well as the outflow through this nuclear envelope. This found on the outer membrane. But on the inner membrane, that the inner side of the inner membrane, there's a compact network of intermediate filaments, a cytoskeletal elements. That's a inside inner side of the inner membrane is having a network of intermediate filaments, and it supports and maintains the nuclear envelope during the interphase, and this is known as nuclear lamina. This network of intermediate filaments on the inner side of the inner membrane, which is meant to maintain the nuclear envelope and to support the nuclear envelope during the interphase. See, now it is more clear, which all of you can clearly observe. See, this is the, just you can see the cursor, this is the outer membrane, sorry, which is the outer membrane, and this outer membrane is continuous with the endoplasmic reticulum. This is the endoplasmic reticulum, and on the surface of the outer membrane you can see openings which are known as nuclear pores. But on the inner wall of the inner membrane, there are two membranes, the outer membrane and the inner membrane. On the inner wall of the inner membrane, you can see a network of intermediate fibers, and they are forming the lamina of the nucleus or nuclear lamina. So, nucleus contents are located in the nuclear lumen, and this is surrounded by a double membrane, that is the nuclear envelope, which consists of outer membrane as well as the inner membrane. Again, to repeat, the outer membrane is continuous with the endoplasmic reticulum, which you can clearly see here, and this nuclear membrane contains number of pores for the movement of the molecule across the membrane, but this membrane, these openings or nuclear pores are highly regulated by protein complexes. Inner membrane has a protein lining called nuclear lamina, and this nuclear lamina binds to chromatin and other nuclear components. So this is nucleus is the most important organelle in the cell. In mammalian cell, except RBC, all cells contain the nucleus. But in case of the prokaryotic cells, there is no membrane, so that's why there is no distinct nucleus. That is the major key character to differentiate between prokaryotic cell and eukaryotic cell. In eukaryotic cell, we have a distinct nucleus, whereas in prokaryotes, there is no distinct nucleus. So, the major structure of the nucleus includes nuclear envelope, nucleolus, then nuclear matrix, or what you call nucleoplasm, inside which you can see chromatin and nuclear lamina. These are the parts. What are the major functions of the nucleus? One is the inheritance, because they maintain the genetic continuity of generation by the replication of DNA and proliferation of the cells. They are very, very important for inheritance of the characters from parents to offspring. And even they are responsible for the development. They regulate the cell differentiation. Yeah, what do you mean by cell differentiation? Initially, when the young cells are formed in the bone marrow, the role and fate of those cells are not decided. It is decided by the nucleus, which cell will develop into what type of organ or tissues. That is the regulation, cell differentiation. It all depends on the gene expression or the genetic expression. So this is the electron microscope structure of the nucleus. So here you can see the chromatin, nuclear envelope. First, let us start understanding the nuclear envelope from the border, from the periphery. What is the outermost region, which is the nuclear envelope or nuclear membrane? I have told you it's a phospholipid bilayer which packs the nucleus, which surround the nucleus. This nuclear envelope that separate the DNA from the the cytoplasm, and it provides a stable internal environment for all the genetic material. Thus, it protects the DNA from damages, and it also separates the replication of DNA from the translation of DNA spatiotemporally. It separates the replication of DNA from the translation of RNA, because I don't know whether you have studied or not, the protein synthesis has different stages. Translation is one such thing, and it separates this translation from the separation, replication of DNA. The chromatins in which you can see the DNA, they are anchored on the nuclear envelope. Then on the nuclear envelope, there are pores which are nothing but the channels for the exchange of the molecules or substances. So these are the important functions that are assigned to nuclear envelope. Because of the nuclear envelope, there are two distinct fluids. One is cytoplasm outside the envelope. Inside, it is known as the nucleoplasm. This nuclear envelope is a bilayer membrane. That is outer membrane as well as the inner membrane. In between these two membranes, there is a space what is known as perinuclear space. There are two membranes. The space which is in between these two membranes, it is called as perinuclear space. There are nuclear pores on the membrane that are linked with the plasma. Outer membrane communicates with the plasma, inner membrane communicates with the nucleoplasm. Ribosomes are attached to the outer wall of the outer membrane, because outer wall of the outer membrane is continuous with the endoplasmic reticulum, and ribosomes are studded on this endoplasmic reticulum. Even the perinuclear space is also linked with the endoplasmic reticular space. There are some intermediate filaments which are of the size of 10 nanometers. They attach to the outer nucleus, outer nuclear membrane. So, the location of the nucleus and endoplasmic reticulum are not movable. That means These intermediate filaments are responsible for anchoring the nucleus in its position. The unmovable locations are convenient to the co-function of nucleus as well as endoplasmic reticulum. The nuclear lamina. What is nuclear lamina? A meshwork of intermediate filaments. They are attached to the inner side of the inner nuclear membrane, are responsible for stabilizing the shape of the nuclear membrane. This nuclear lamina, which is a meshwork or network of intermediate filaments which are attached to the inner wall of the inner membrane, helps to stabilize the shape of the nucleus. See, these are the nuclear membrane, the double layer. There is a small space which you can clearly see here, that is the nuclear perinuclear space. One is the outer membrane and another one is the inner membrane. Outer membrane is continuous with the endoplasmic reticulum, whereas the inner membrane, it is having a network of meshwork of intermediate filament, that is what you call nuclear lamina. These are all nucleoplasm in which you can see the chromatin fibers. And on the surface of the nuclear envelope, you can see minute openings, pores. Can you make out here? And these are, wherever there are arrows are shown, these openings are known as nuclear pore complexes. Nuclear pores which are regulated by protein complexes. See, this is the nuclear membrane that has been observed under the electron microscope. See, this is the endoplasmic reticulum. I have told you the outer membrane is continuous with the endoplasmic reticulum. This is what is known as perinuclear space, the space between the outer membrane as well as the inner membrane. So here, this is the nuclear complexes, pore complexes, where there are openings. Here one more is here. Here also you can see. These are the nuclear openings or pore complexes. They are functionally similar to that of the nuclear membrane, similar to that of the plasma membrane. If you observe that the nuclear membrane through the scanning electron microscope, you will get such a type of pictures. All these openings are nothing but the nuclear pores. See, innumerable number of such nuclear pores on the surface. And this is the nuclear lamina observed under the electron microscope. It's a meshwork of intermediate fibers just below the inner membrane. That is what is known as nuclear lamina. This is the electron micrograph. So now, let's try to understand about the nuclear membrane. See here, these are the, what is important for us is the nuclear pore complexes. See, they are guarded by complex proteins. This is the lamina. This is the chromatin. This is the space, perinuclear space. This is the inner membrane, this is the outer membrane. Outer membrane is continuous with the endoplasmic reticulum. Okay, now let's try to understand the functions of the nuclear lamina. There is a meshwork of intermediate fiber on the inner wall of the inner membrane. This is responsible for the shape of the nucleus. So there is the shape, it will not allow the change in the shape of the nucleus. And even if you use even some of the salt solution, detergents, on the nucleus, the still it presents a shape to the nucleus. The it is associated with the assembly of chromatin and nucleus. Assemblage of chromatin. The shape of the nuclear lamina can change only during the cell proliferation phase, that is the G1 phase. If you remember the cell cycle, G1 phase, S phase, G2 phase. So only during G1 phase the shape of the nuclear lamina may change. At the end of G1 phase, the nuclear lamina will be phosphorylated and nuclear envelope will disappear after that one. All types of lamina, they get phosphorylated and assembled again to form the nuclear lamina immediately after the cell division. See, this is the meshwork. Then, nuclear pores. Nuclear pores are guarded by protein complexes. And the nuclear proteins are synthesized in the plasma, and then it will be imported into the nucleus by these pores. Actually, the proteins what are required for the nucleus, that is produced in the cytoplasm. Then such produced cytoplasm, the proteins are to be sent inside. The RNase and ribosome subunit, they synthesize the protein, which all of us have studied while discussing the the ribosomes. So now the nuclear pores, it should come inside through the nuclear pores. If you observe individual nuclear pore, it is composed of 50 different nucleoproteins, and they form nuclear pore complexes, that is known as NPC. Usually a mammalian nucleus contains as many as 3000 nuclear pores. The number of nuclear pores in a cell in the nucleus depends upon the activities. More the activities, there will be more nuclear pores. For example, the ovum of the frog contains 37.7 into 10 raised to 6 nuclear pores. But the matured cell contains 150 to 300, because frog ovum is more active, very highly active. The structure of any nuclear pore includes a cytoplasmic ring located on the cell plasma. It has 8 filaments and a nuclear ring, which are also the extensions of 8 filaments. Then there are transporters and the spokes. Probably it will be clear. See, the nuclear pores are almost octagonal in shape. This is the electron microscope photograph. See, they are showing octagonal shape. Probably next. Okay, we will not go to this. Yeah, this is almost octagonal because there are 8 spokes. These are the basket filament. These are the spokes. See here, there are outer ring and this is the inner ring. Outer ring and the inner ring. And here you can see these are the spokes, 8 spokes. And these are the cytoplasmic ring. and in the center there is a plug, central plug, and this entire structure is known as nuclear basket. This is the terminal ring. So this is the structure of the nuclear pores, and they are octagonal in shape, and they are guarded by these protein complexes. Now you can see they are not free openings but guarded by protein complexes. See here the different types of protein complexes, how they are arranged. We need not go to the. There are almost approximately 30 different types of proteins are found in these pores. See here, there are more than 30 different, and basic subunits repeat 16 times. The role of these protein complexes is to regulate the flow. They decide what to come inside and what to go outside. It may be along the concentration gradient or it can be against the concentration gradient. That is the nuclear pore complexes. After this nuclear envelope, let us move on to the cytoplasm, study of the cytoplasm of the nucleus. I have told you the cytoplasm of the nucleus is known as nucleoplasm. Nucleoplasm is also known as karyolimb. It's also called as karyolimb. It is nothing but the space inside the nucleus. Space inside the nucleus contains, like the cytoplasm, many dissolved substances and suspensions. Like the cytoplasm, there are many dissolved substances and suspensions. Dissolved substances are usually mineral solutes. Dissolved substances are usually mineral solutes. Suspensions include different kinds of enzymes, chromatin network, and nucleoli. Similar to the cytoplasm of the cell, the nucleus contains nucleoplasm. It is also known as nucleus sap or karyolimb. Nucleus sap or karyolimb. So, it's not wrong if you say nucleoplasm is one of the types of protoplasm, and it is enveloped externally by the nuclear envelope. So, nucleoplasm gets separated by the cytoplasm by means of this nuclear envelope. The nucleoplasm is highly viscous liquid, because within this liquid there are chromosomes and nucleoli. In the nucleoplasm you can see many substances like nucleotides, enzymes get dissolved in the nucleoplasm. Suppose if you filter this nucleoplasm, the soluble only liquid portion of the nucleoplasm is called as nuclear hyaloplasm. If you remove all from the nucleoplasm, if you remove all the suspended material, what remains is a liquid portion of the nucleoplasm, and that liquid portion of the nucleoplasm is called as nuclear hyaloplasm. Like cytoplasm, the nucleoplasm is also made up of mostly water, mixture of various molecules and dissolved ions. It is highly gelatinous, sticky liquid. Gelatinous and sticky liquid that support the chromosome and nucleoli. The main function of the nucleoplasm is to serve as a suspension substance. It functions as a suspension substance for the organelles inside the nucleus. What are the organelles? Chromatins and nucleolus. And it also plays an important role in the transport of the nuclear materials. Which are vital to the cell metabolism and function. Transportation of these vital materials is also through this nucleoplasm. As I have already told you, there are many substances such as nucleotides, enzymes, they get dissolved in this nucleoplasm. Now, in the nucleoplasm, we have to study two organelles. One is the chromatin and another one is the nucleolus. I am not discussing in detail about the chromatin because in the next class we have to discuss about this only. But still, as a cursory glance, I will discuss only the important things of this chromatin. What is chromatin? Chromatin is nothing but combination of DNA and histone proteins. That is DNA and special class of protein, what is known as histone proteins, they are together called as chromatin. Histones are those proteins which are having high percentage of alkaline amino acids. Histones are those proteins which have high percentage of alkaline amino acids such as arginine and lysine. So a chromatin is nothing but DNA plus histone. You call it as chromatin. Chromatins show two reasons. One is it's a long fiber thread-like structure. Naturally it shows two reasons. One is euchromatin portion and another one is heterochromatin. Euchromatin, they are lighter, thinner and less dense. They are lighter, thinner and less dense, and it is the active part of the chromatin. It is the active part of the chromatin with uncoiled genetic material, remember. They are lighter, thinner and less dense. So even if you stain, they take light staining. But it is the active part of the chromatin. Another one is the heterochromatin. Contrary to euchromatin, the heterochromatins are dark, thick and very dense. Dark, thick and very dense, and genetic material is highly coiled. Genetic material is highly coiled, condensed and genes are inactive. So genes are active in the euchromatin but inactive in the heterochromatin. So this chromatin was named by Fleming in 1879. Then the chromosomes term was named by Waldeyer in 1888. Now the question is, what is the difference between chromatid and chromosome? Chromatid and chromosomes are same substances with different shapes. Shape presentation in different cell cycle phase. Chromatin and chromosome, they are one and the same, but they attain different shapes. When the cell is in a non-dividing phase or in the interphase, we call it as chromatin fiber. If the same chromatins condense, become short and thick, which happens during the cell division, mitosis and meiosis, then you call it as chromosome. That's the only difference. The chemical composition of the chromatin, if you look into them, they compose of DNA, deoxyribonucleic acid, and mainly histones, some amount of non-histone proteins, and some RNA at the ratio of one part of DNA, one part of histone, and 0.0.5 portion of non-histone proteins and RNAs. All of you know DNA is the carrier of the genetic information. DNA sequences can be sorted as three types: non-repeated fractions, moderately repeated fractions, and highly repeated fractions. Of course, the DNA that we will discuss when we discuss the DNA is available in three forms: B form, Z form, and A form. That is known as aDNA, bDNA, and zDNA. See, these are the forms of DNA: aDNA, zDNA, and bDNA. DNA is because DNA is the major component of the chromatin. So the chromosome DNA contains three basic sequences. One is autonomously repeating DNA sequences. Second one is the centromere DNA sequences, and the third one is the telomere DNA sequences. Again, I will discuss this later, not now. These are, I will avoid it right now, because when I take up in the next class, we have to discuss about this DNA only. After DNA, the another major component is the histone. Histones are positively charged proteins containing arginine, lysine, alkaline proteins. Even histones can be sorted as two types: highly conserved core histones and non-conserved linker histone. Highly conserved core histones and non-conserved linker histones. Highly conserved core histones are named as H2A, H2B, H3, and H4. Non-conserved linker histones including they have only H1 protein. Most of the amino acids of the core histone, they interact with DNA and other histones. Whereas the linker histones, H1, is the one, as the name indicates, linker. It helps in linking the highly conserved core histones, that is H2A, H2B, H3, and H4, with the other histones. In between this, you can see this linker histones. Then non-histone proteins. Even there are some amount of non-histone proteins are there. They are negatively charged and acidic proteins, which are made up of large number of aspartic acid and glutamic acid. They are synthesized in the cell cycle, in the entire phase of the cell cycle. But remember, histone proteins are synthesized only during synthetic phase of the this cell cycle. These non-histone proteins are very essential because they recognize the specific DNA sequences. The non-histone proteins have certain role, like they help the DNA molecule to be pleated and form different structural domains. Then help to start DNA replication, and they also regulate transcription and gene expression. There are 23 pairs of chromosome in the human nucleus. If you open and extend the DNA molecule in each chromosome, each DNA molecule will be around 5 centimeters long. If you link all the DNA molecules, it will measure about 1.7 to 2 meters long. But remember, the diameter of the nucleus itself is less than 10 millimicrons. So the genome information is packed into such a tiny structure, 1,000 times smaller than a dot. Now, the next one is, if you take a chromatin fiber and treat it with some nuclease enzyme, non-specific nuclease enzyme, DNA gets fragmented, and each fragment will be 200 base pair. One base pair, base pairing, adenine, guanine, thymine, cytosine, just you recollect. For every 200 base pair, they break. Then if you treat this DNA with that enzyme, you will get degenerated fragments of DNA. This was done by Korenberg. If you hydrolyze this DNA, that ends in a structure what is known as nucleosome. Nucleosome is the core particle which are wounded with the histone proteins. Each nucleosome, if you see, it has one histone core and an linker histone. So this is what you call the nucleosome. See here, if you structure of a nucleosome, see this is the histone core, octameric histone core, and these are H1 histones or linker histones. How? A long thread of DNA has been formed, and each unit, see the histone core and the DNA along with linker histone, and this is the DNA, and the central one which you are seeing almost yellow color, that's the what you call the histone core. This histone core, DNA, and the linker DNA, linker histone, you call it as one nucleosome. So this also I will take up in. So yeah, the DNA field, this is the nucleosome. How it appears under the electron microscope. So what has been done actually in the chromosome? The DNA is packed. So DNA, it is folded in the, see, this is the short region of DNA that it is wounded with the beads. Beads go for again the foldings, further foldings, and ultimately they are represented in the form of a chromosome. And about this heterochromatin, euchromatin and all these things, I will take it in the next class, because in the next class I have to take the discussion of this DNA, Just we have one thing left, nucleolus. I will finish the nucleolus and then I will move on to the uh, so could you hear ಕೇಳ್ತಾ ಇತ್ತಲ್ಲ